Greetings. I'm Greg Marcus, a cardiac electrophysiologist and professor of medicine at UCSF, and delighted to tell you all about our CRAVE trial, which stands for Coffee and Real-Time Assessment of Atrial and Ventricular Ectopy. This was our attempt to rigorously assess the acute effects of coffee in ambulatory individuals using a randomized crossover study design. So we enrolled 100 volunteers and fit them with a accelerometer, the Fitbit Flex 2, that could record step counts as well as sleep duration. We fit them all with a continuously recording EKG device using the Zeo patch made by iRhythm. We also collected DNA for genotyping of caffeine metabolism variants, and fit them with a continuous glucose monitor. And then they were randomly assigned via a mobile app built on our NIH-supported Eureka digital research platform to either avoid or consume coffee on a day-to-day basis. And we were able to validate whether they followed those instructions in several ways. We first reimbursed for all coffee as long as those participants provided a date-stamped receipt, whether they were supposed to have consumed coffee or not. We also used a geolocation tracker and actually were able to ascertain visits to coffee shops. And then we asked our participants every day whether they really consumed coffee or not. So what we found, comparing days when people were randomly assigned to consume or avoid coffee, was no difference in premature atrial contractions. In the per protocol analysis, meaning if they actually consumed coffee independent of the intention to treat, more coffee consumed was associated with significantly less supraventricular tachycardia episodes. In contrast, in both intention to treat as well as per protocol, whether it was random assignment to consume coffee or the actual number of drinks consumed, that was associated with more premature ventricular contractions, and that was highly statistically significant. Interestingly, looking at the genetic results, those who metabolize caffeine more quickly seem to experience a more potent response, meaning faster caffeine metabolizers demonstrated more PVCs when exposed to coffee. The most robust results came from the Fitbit analyses, where we found that, again, whether it was randomized assignment or per protocol, coffee exposure was associated with significantly more step counts, on average about 1,000 more step counts per day on those days associated with coffee consumption per random assignment. At the same time, those days when individuals were randomly assigned to consume coffee were associated with substantially less sleep that subsequent evening, on average about a half hour less sleep. And again, when we looked at the genetic analyses, as expected, actually, those who metabolized caffeine more slowly experienced a heightened relationship here. So those who genetically were predisposed to metabolize caffeine more slowly experienced more reductions in sleep when they consumed coffee. We were unable to identify any differences in serum glucose. So in thinking about the implications of these findings, so overall, we find that the physiologic effects of coffee are quite complex, even when we just look at acute or nearly immediate results. These data add to the growing evidence that those with supraventricular arrhythmias or atrial fibrillation or at risk for those should not necessarily avoid coffee. We can find no evidence that coffee increased the risk for those arrhythmias, and in fact, some suggestion that coffee may reduce the risk of such arrhythmias. In contrast, those prone to premature ventricular contractions or ventricular arrhythmias may benefit from coffee abstinence, but it does appear that there are differences based on genetic variants that are common in the population. Coffee may enhance overall physical activity, but this needs to be weighed against the risks associated with reduced sleep. And finally, those who metabolize coffee more slowly seem to be at greatest risk of coffee-induced reductions in sleep. Thank you very much.